pollination biology and pollinator communities of the endangered Willamette Daisy in the Oregon Prairies.
Okay, here we go. Great. Let me know if you can't see my screen. I'm going to go ahead and jump right into it. I'm going to tell a story about a study we're doing about the role of pollinators in the conservation of an endangered plant in Oregon. My name is Scott Harris. I'm with Institute for Applied Ecology, and I only represent a team of people working on this project. The others are included there: Tom Kay, Christina Mitchell, David Kappert from the same organization, and then Susan Waters from Quamish Eco Research. So just a real quick plug about our organization, Institute for Applied Ecology. We're a nonprofit based in Corvallis, Oregon, in the United States, which you can see there. And our mission is to conserve native species and habitats. And the way we do that is through active habitat restoration, which includes activities from planting to prescribed fire, herbicide application, and management of critical habitats. We have a conservation research program where we monitor the status of threatened and endangered species, mostly plants. And then we also conduct research in the efficacy of restoration practices, ecological education program. Then we also have a very active farm that provides plant materials for a lot of our work. An example of a recent success, we hope is a success, as we worked with this endangered butterfly species, Fender's blue butterfly, with partners well over 10 years, and it's currently being considered for downlisting from endangered to threatened. So that's an example of some of our work. But this is BeeCon, so we're here to talk about bees. And please excuse my including of flies in this slide. But these are just some of the characters we're working with in this current study. So, we have, for example, an example of a sweat bee here, and a hoverfly, carpenter bee, and then coniferous. Many of the pictures you see in my presentation, nearly all of them are from David Kappert, both pictures on flowers and also 3D stacked pictures from pinned specimens. If you're curious about the photos, I can connect you with David. So a little bit about the plant we're working with, Willamette Daisy, Erigeron decumbens. It's in the Asteraceae family. Throughout this talk, I'll probably be referring to it as ERDI. That's the code for it. It's listed as endangered on the U.S. Endangered Species Act, and it's endemic to prairies in the Willamette Valley of Oregon, the map I showed earlier. It's a perennial plant. It has clonal growth, as you can see in that picture at the right, and that's typical habitat in oak savanna prairie habitat. But the clonal growth is in very small clumps. Otherwise, it's very dependent on seed for reproduction. Another interesting thing is that the flower head has between 100 and 200 florets, and they serially mature from the outside in, which you can see in that picture. You can see some that are totally mature, all florets compared, and this is the same plant, or some that aren't. So each plant goes through that maturation stage of their florets in a period of about 3 weeks. So when we're talking about conserving an endangered plant, we need to think about the very small scale, the plant biology specifically, and also, how it fits in the community. In terms of plant biology, we need to know about the structure, growth, and development of the plant, how maybe it evolved and maybe its genetic adaptations to its environment. Also about the soils, the nutrient needs, and environmental adaptations of the plant. Additionally, we have to understand the community aspects of plant conservation. It's not just one plant existing in isolation. It interacts with a whole suite of other taxa, and that can be from competition and facilitation within the plants. And then if we're talking about other trophic levels, for example, herbivory and other interactions. And then finally, another component, which is what we're going to talk about here, is pollination, which isn't necessarily always considered in plant conservation. So that's the gap we're trying to fill with this particular plant. So our main question is what is the role of pollinators in the conservation of Willamette Daisy? Or do they play a key role that ensures that we can maintain this plant on the landscape? And sub questions under that are, well, first of all, are pollinators even needed for this plant? And if they are, then which pollinators are there that visits this plant that we can maybe make sure we engage in practices that conserve the pollinator community as well? That's important for the plant. So the way we did that, again, this is the Willamette Valley in Oregon, just south of Portland. We had 8 prairies in the Willamette Valley that we visited for this study. We identified patches of Erde, although that is a picture, that's not a picture of Erde, that's, I believe, lupin in the picture there, but we identified patches of our target species, Erde, in each of these prairies. And for each patch, we spent 6 hours of observation and observed pollinators visiting the plant and where they went afterwards, and also collecting and identifying the pollinators that visited this plant. And like I said before, the plant matures over about a 3-week window. So that 6 hours was spread out throughout that 3-week window for each patch. Here's an example of some of our study sites. So as you can tell, they range from very pristine prairie communities, prairie habitats, to humid modified. But we did have our target species at each of these sites. These are just examples of some of them. In addition to collecting pollinators and observing pollinators at patches of Willamette Daisy, we also did a pollen supplementation study. This is a pretty fascinating project where we had honeybees that we attached to toothpicks, and we passed the honeybee with our bee stick across a Willamette Daisy plant, and then transferred that pollen to other plants. So that was one way of artificially supplementing pollen to individual flower heads or plants of Willamette Daisy. In addition to this, we also had Willamette Daisy that were open, that were not supplemented, that were just experienced the native or natural pollination of the community that was there. And another component of the experiment, we also had caged plants, so that were totally closed to any pollination, whether supplemented or artificial, to see if Erde could self-pollinate. Then some of our results. In this graph, so seed set is on the left, and that's the percent of filled seeds. So if you have 100 seeds coming out of a single Erde plant, if seed set's 50%, that means half of them are filled. The 2 lower bars are for non-supplemented pollen. They're for the caged and the non-supplemented pollen. So seed set is really low. This shows us that Erde does very rarely self-pollinates. And we don't even know if those plants that self-pollinate can even create a progeny that successfully reproduces and adds to the population of the plant. That's a next step of our study. But then the bars on the right are from pollen supplementation. I don't want to go too much in the weeds on this part, but we also did a study on that tested outbreeding depression, which we didn't find in this plant either. But the point here is that, yes, Erde definitely needs pollinators. There's quite a significant increase in seed set when pollen is supplemented. Also at the site level, this is another way of looking at it. So this is a seed set again on the left side, on the left axis. And the purple boxes are the flowers that were open to natural pollination, so they experienced whatever pollinator community was there. The orange boxes are the ones that we supplemented with our bee brushes. So you can see at every site, supplementing pollen increased seed set of the plant. And that was for 2021, excuse me, and this is 2022. So it ranged from 1.1 times to up to 6 times increase in seed set by supplementing with pollen. So first we found that, Erde needs pollinators to have seed set. And then what this is showing here is that Erde is pollen limited. There's something happening out there, or maybe that's not right the way to put it. The right way to put it is that the plant isn't receiving its full potential from the pollinator community. And maybe the reason it's not reaching its full potential, there could be many reasons for this. So this is about the wild pollination. There's open pollinated plants. The ability of a pollinator to successfully pollinate a plant is dependent on a number of things. One is, for example, just trait matching or anatomical matching. Is for example, tongue length or different features of the pollinator, do they match the plant so that that pollen is successfully transferred that can aid reproduction? Foraging behavior of the pollinator is also important. Its abundance in population dynamics, how they interact within a network, a plant pollinator network. Also constancy and stigma contamination. Stigma contamination is, imagine a field full of flowers of which some are early. If pollinators are visiting all these flowers, getting all pollen from different flowers and then visiting early, the stigmas of early might be contaminated with pollen that won't work for its reproduction. So that's another concern. But looking at constancy and stigma contamination, we did watch pollinators on Willamette Daisy, and we found that they were highly constant on Willamette Daisy. So if a pollinator visited Willamette Daisy, 97% of the time they would go to another Willamette Daisy or the same Willamette Daisy as opposed to other species. So that was a good sign. So the next part of the study was which pollinators visit early. I grouped these into very broad categories, as you can see. But the most interesting thing is the difference between flies and bees. So 2019, 2020, flies and bees are going back and forth. But then boom, in 2022, we had quite a change in the proportions of flies compared to bees. This is because we had a very wet, cool spring, which greatly impacted the bee community in these prairies. So it'll be fascinating to see the long-term results of this season on not only this plant, but on the plant community and the pollinator community as well. In terms of bees, so I had those pies that showed flies, bees, wasps, etc. Now we're just looking at the bees. This is just a collection of the bees that we found visiting Willamette Daisy. This is an example of Apidae, and then Halictidae example, mason bees, Megachilidae, and then Andrena. So these are just some of the examples of the most abundant family of bee pollinators visiting Willamette Daisy. And then I couldn't get away from this presentation without saying something about the flies. The most abundant fly ended up being Syrphidae. Further work is, of course, plant pollinator networks. We've probably all seen slides like this. And I just wanted, there's nothing to really say about this except that this is future work for our study. As I mentioned before, Willamette Daisy is a plant that exists within a community of other plants and a community of pollinators, and its role, its space in that network is really important in how its future reproduction and population stability. Another question is about pollinator effectiveness. What about all those flies? Are they actually providing any pollinator services? So what we hope to do is potentially, if it's not too much work and within our means, to actually assess the effectiveness of individual pollinator taxa. And then in conclusion, we found that Willamette Daisy needs pollinators and it is pollen limited. Stigma contamination isn't really an issue because we found high constancy for the insects visiting Willamette Daisy. The pollinator community varies quite a bit by site and year. And the many questions remain include, how effective are not just the flies, but all different types of taxa at pollinating this plant. And then do we need diverse pollinator communities or potentially diversity in pollinator traits? If different species of pollinators go in and out of the community, if those traits that they have that helped pollinate Willamette Daisy are existing, then maybe we should focus on traits as opposed to species diversity.
Hey, Scott, we're a little bit over 14 minutes. So do you want to try and answer any questions?
Yeah, and there's my thank you slide.
So Rowan has asked, since this species seems to be perennials, did the pollen limitation study consider the same plants each year?
Sorry, could you ask the question again?
Did you study with pollen limitation the same plant, i.e. the same stems or clumps every year to see if there was some consistency in that particular individual?
No, because these were in situ. They were actually on site in the prairies. However, we did take the seeds from, for example, 2021, and we are growing those out. So we'll do further measurements and assessment of, the reproductive capacity of those plants that were supplemented. But each year it is a different number of individual plants that are supplemented because they are in situ on site.
Great, thanks. And we're going on